Good afternoon, and welcome to Session 5 on Best Practices for Submitting Formulation Assessment Requests and Avoiding Information Requests. Our first presenter will be Lieutenant Commander Elizabeth Kim, who is a Control Correspondence Coordinator for the Division of Filing Review and the Office of Regulatory Operations in OGD. In her role, she reviews and responds to inquiries submitted by generic drug manufacturers or associated industry on a specific element of generic drug development. Prior to joining the FDA in 2015, Lieutenant Commander Kim worked as a family nurse practitioner, providing primary care in various clinical settings. Our next presenter will be myself, Lieutenant Commander Phung. I am currently a supervisor in the Division of Filing Review in the Office of Regulatory Operations in OGD. I lead teams responsible for reviewing ANDAs and control correspondences, and have collectively over 10 years of experience in varying capacities in both the FDA and the United States Air Force. I will also be the moderator for this session, and we will be accompanied by Lieutenant Commander Elizabeth Kim, our first presenter, Drs. Julia Lee, Melissa Mannion, and Bryan Newman as panelists. Dr. Julia Lee is the Deputy Division Director of the Division of Filing Review, Office of Regulatory Operations in OGD. Dr. Lee has over 10 years of experience in the filing review of abbreviated new drug applications submitted to the agency. Prior to working at the agency, she was a retail pharmacist at Walgreens. She has also worked in the Chesapeake Atlantic node of the Pediatric Emergency Care Applied Research Network as a clinical research assistant. Next, we have Dr. Melissa Mannion, who is a regulatory counsel with the Division of Policy Development in the Office of Generic Drug Policy. In her role, Dr. Mannion works to develop and implement new and improved policies, guidances, regulations, and other policy documents to support the approval of high-quality generic drugs on the first lawful approvable date. She serves as a policy subject matter expert on a range of complex issues pertaining to generics, including bioequivalence requirements, pre-Hatch-Waxman ANDAs, inactive ingredient sameness, disclosure concerns, and importation. Dr. Mannion has more than 15 years of experience encompassing public health policy research, regulatory and legislative affairs, pharmaceutical purchasing and distribution, professional affairs, and retail pharmacy operations. And lastly, we have Dr. Bryan Newman, who is a lead pharmacologist and team lead for inhalation and nasal drug products in the Division of Therapeutic Performance 1 in the Office of Research Standards in OGD. Dr. Newman's work focuses on developing product-specific guidances and addressing control correspondences, citizen petitions, consults, and pre-ANDA meeting requests. He also serves as a project officer and contracting officer's representative for the regulatory science research initiatives related to inhalation and nasal drug products. Thank you for joining us today. We hope that the presentations will be informative.
Hello, my name is Elizabeth Kim. I am a Controlled Correspondence Coordinator for the Division of Filing Review within the Office of Generic Drugs at the FDA. Today, I will be presenting on the best practices for submitting formulation assessment requests and avoiding information requests. I'll be also sharing some tips for submitting a proposed formulation table. Today's objectives are learning how to submit high-quality formulation assessment requests, how to avoid information requests, and understanding do's and don'ts when submitting a formulation table. This slide focuses on the information requests related to controlled correspondence. A controlled correspondence is a pathway a generic drug manufacturer or related industry could utilize to request information on a specific element of generic drug product development. A request for qualitative and quantitative formulation assessment is one type of controlled correspondence inquiry that could be submitted to the agency. An information request is sent by the agency in the course of reviewing a controlled correspondence when more information is needed to conduct an assessment. When reviewing a Q1, Q2 formulation assessment inquiry, omission, discrepancy, or ambiguity in the formulation table can trigger an information request. Unlike abbreviated new drug applications, a Q1, Q2 formulation assessment control correspondence inquiry contains brief information of the drug product and no more than 3 proposed formulations. Hence, an unclear and ambiguous proposed formulation may lead to an inaccurate assessment due to misinterpretation, or trigger an information request, or lead to an unable to comment response. Therefore, it is important to submit a high-quality formulation table that is clear with adequate details. Now we'll go over some tips on submitting proposed generic formulations for assessments. The function of all ingredients should be included in the proposed formulation table. This is especially important when the agency is assessing certain allowable differences permitted under 21 CFR 314.94A9, Romanet 3-4. Parenteral drug products must contain the same inactive ingredients in the same concentration as the reference-listed drug. However, specific changes are permitted for preservative, buffer, antioxidant, provided that the applicant identifies and characterizes the differences and provides information demonstrating that the differences do not affect the safety and efficacy of the proposed drug product. Ophthalmic and otic drug product also must contain the same inactive ingredient in the same concentration as the reference-listed drug. Exception excipients that may differ from the reference-listed drug include preservative, buffer, tonicity agent, and thickening agent. Please note that for ophthalmic drug products, FDA has determined that, as a scientific matter, any qualitative and quantitative differences from the reference-listed drug should be accompanied by an appropriate in vivo BE study or studies. This includes differences in the exception excipients. The agency will not be able to assess allowable differences when there is an omission of the function of the ingredients in the formulation during the controlled correspondence formulation assessment review. Hence, the function is an important part of the generic drug product formulation. Next, when submitting a Q1, Q2 formulation assessment inquiry, please provide the proposed formulation table in all relevant units, including milligram per mil, percent weight per weight, percent volume per volume, and percent weight per volume as applicable. The same units of measurement as the reference-listed drug is generally used to conduct Q1, Q2 formulation assessment during the controlled correspondence review. Next, it is important to clearly indicate when an excipient will not remain in the final drug product. In this example, the control correspondence submitter clearly indicates that water is removed during lyophilization. Continuing on, please ensure that the proposed formulation is clear and unambiguous as possible. In the proposed generic drug formulations, please include hydration state, grade, purity, and viscosity as applicable. The description of each excipient can be included within the ingredient column of the formulation table, or you could utilize a footnote to describe an excipient. Common issues that we noticed during the Q1, Q2 control correspondence reviews are as follows. 1. Omission of description of ingredients. 2. Ambiguous description of ingredients, which could lead to misinterpretation during the review. And 3. Discrepancy between the description of an ingredient listed in the formulation table and the footnote. This is an example of an omission. In the first example, the hydration state of edetate disodium is not listed. According to the USP monograph, edetate disodium comes in anhydrous and dihydrate forms. Without the hydration state, the agency is unable to determine if the proposed amount of edetate disodium is quantitatively same as the reference listed drug. The regulation requires certain drug products to be qualitatively and quantitatively the same as the reference listed drug product. Hence, in order to assess the quantitative sameness in this case, the hydration state is needed. The second example clearly indicates that hydration state of edetate disodium is dihydrate. The component column lists edetate disodium dihydrate accompanied by a footnote stating that edetate disodium dihydrate 1.25 milligram listed in the composition table is equivalent to 1.13 milligram of edetate disodium anhydrous. If there is any water correction of edetate disodium, the footnote is clear as to the amount of edetate disodium in the final drug product. This is an example of a discrepancy. In the first example, there is a discrepancy between the listed hydration state of sodium acetate in the formulation table and the footnote. The formulation table indicates that hydration state of sodium acetate is trihydrate and the amount listed in the table is 0.22%. However, the footnote indicates that 0.22% sodium acetate anhydrous is equivalent to 0.365% sodium acetate trihydrate. It is unclear if the amount listed in the first formulation table is trihydrate or anhydrous due to the discrepancy between the formulation and the footnote. In the second table, the amount of sodium acetate trihydrate is 0.22%, which matches the footnote. In the next few slides, we will go over a few examples of both clear and unclear descriptions of excipients. When the agency is conducting a Q1, Q2 assessment of a proposed generic drug formulation in a controlled correspondence inquiry, the agency does not have any information other than the proposed generic formulation. Hence, it is important to submit a clear formulation table to avoid an information request and ensure timely review. The first table is an example of an ambiguous description of hydrochloric acid. Note that hydrochloric acid, as listed on the formulation table, is absent a declared normality. The footnote to the same entry indicates hydrochloric acid will be added as 1 normal hydrochloric acid. This is ambiguous because it is unclear whether 6.1 milligram per ml hydrochloric acid, as listed in the composition table, represents a neat concentrated amount of hydrochloric acid or 1 normal solution of hydrochloric acid. One could potentially interpret that 1 normal hydrochloric acid is used during the manufacturing process, but the quantity listed in the composition table is a neat concentrated amount. Let's look at the two clear examples. The first table under the clear examples also has a footnote. It states that the concentration of hydrochloric acid as listed on the composition table represents 1 normal hydrochloric acid solution. This is clear that hydrochloric acid 6.1 milligram per milliliter listed represents 1 normal hydrochloric acid. In the last example, the formulation table does not have a footnote. However, it is clearly indicated in the ingredient column that 1 normal hydrochloric acid is used. As mentioned, unlike ANDA submissions, the controlled correspondence inquiries do not contain additional information other than the proposed drug formulation. Hence, it is important that each ingredient is described as clearly as possible for a timely and accurate assessment. We are continuing on with ambiguous descriptions. Please take a look at the first table. The ambiguous example of benzalkonium chloride has a footnote that states that benzalkonium chloride is added as benzalkonium chloride 50% solution. It is unclear whether that 50% solution was used in the manufacturing process and the amount listed in the composition table is a neat concentrated amount or 50% solution. Now let's look at the clear examples. If you look at the table in the middle, benzalkonium chloride 50% solution is listed in the component column. The footnote states that benzalkonium chloride 0.3 mg per milliliter, parentheses 50% aqueous solution, is equivalent to 0.15 mg per milliliter of benzalkonium chloride. In this example, it is clear that the amount listed in the quantity column of the formulation table is the amount of benzalkonium chloride 50% solution. Let's look at the last table. Benzalkonium chloride is listed in the ingredient column. The footnote reads, benzalkonium chloride will be added as 50% solution. Final product will contain 0.1 milligram per milliliter of benzalkonium chloride. In this case, it is clear that the quantity column represents a neat concentrated amount of benzalkonium chloride. In this slide, the quantity listed in the formulation table is unclear. In the first example, the quantity of hydrochloric acid is listed Na. In the table in the middle, the quantity of hydrochloric acid is listed with a dash. In both examples, there is a footnote which states hydrochloric acid used for adjusting pH. It is unclear if the amount of hydrochloric acid is not applicable, not used, or used as needed. The controlled correspondence submitter should clarify the meaning of Na or dash listed for the hydrochloric acid in the proposed formulation tables. If you look at the clear example, the last table, the quantity of hydrochloric acid is listed QS, as needed or amount which is enough. This is much clearer and there's no room for misinterpretation. Next, here's an example of ambiguous word use. In the first example, the word dehydrate is listed in a parenthesis under edetate disodium. The use of the word dehydrate is unclear whether edetate disodium is dehydrated, meaning dried and anhydrous, or is a typo for dihydrate. In this case, a footnote may help clarify the meaning. The second table clearly states in the component column that edetate disodium is dihydrate. It even has a footnote stating that edetate disodium dihydrate 1.25 mg is equivalent to 1.13 mg of edetate disodium anhydrous. In this case, there is no room for misinterpretation. Now I'll talk about total fill volume or fill size of parenteral drug products. When submitting Q1, Q2 formulation assessment requests for parenteral drug products, please indicate both the concentration and total fill volume of the proposed generic drug products within the request or in the formulation table. Generic parenteral drug products must contain the same concentration and total drug content per container as the reference listed drug. Please note that per the guidance for industry on controlled correspondence related to generic drug development from December 2020, the agency recommends that formulation assessments for multiple drug products be submitted in separate control correspondences. This includes separate formulation assessment requests for drug products with multiple strength, such as parenteral drug products with different fill sizes, as each fill size is a separate drug product. This slide is on how to submit a proposed generic drug formulation for co-packaged drug products. It is important to include a drug composition for each packaged component for co-packaged drug products. In other words, if a drug product comes in a dose pack, such as a vial containing lyophilized powder and diluent, please include formulations for both the lyophilized powder and the diluent. It is important to provide compositional breakdown, both qualitative and quantitative, for all components of a drug product. Some examples of co-packaged drug products could be a kit containing both lyophilized powder and diluent, a kit that contains two or more separately packaged components, such as a vial with lyophilized powder, syringe 1 containing solution, syringe 2 containing another solution. In this case, three separate compositions of each packaged components are recommended. In summary, when submitting Q1, Q2 formulation assessment requests via the Controlled Correspondence Program, ensure that the proposed generic drug formulation is clear with adequate details so that there's no room for misinterpretation. Please include the function of all ingredients, list all relevant units of measurement for each ingredient as applicable, and list the description of each ingredient, including hydration state, grade, purity, and viscosity as applicable. Also, for parenteral drug products, please be sure to include both the concentration and the fill volume in your controlled correspondence inquiry. Lastly, if you are submitting a formulation assessment request for co-packaged drug products, please submit a formulation for each component of the co-packaged drug product. Thank you for your participation. I'll be back during the question and answer session to answer any questions you've submitted. Thank you.
Good afternoon, and welcome to today's presentation on pathways for receiving FDA's feedback on formulations. My name is Vinh Phung, and I'm Lieutenant Commander in the United States Public Health Service, and also a Regulatory Officer in the Division of Filing Review, Office of Regulatory Operations, and OGD. As an overview, I will be providing background information and elaborate on two particular pathways, control correspondences, and product development meetings. Regulations generally require that certain proposed products be qualitatively and quantitatively, also known as Q1 and Q2, the same as the reference listed drug with respect to certain inactive ingredients. Under 21 CFR Section 314.94 Roman at 3 through 4 are regulations for drugs intended for parenteral, ophthalmic, or otic use. FDA recommends that applicants request a Q1, Q2 assessment for potential formulations prior to submitting an ANDA. One of the pathways to receive FDA's feedback is submitting a controlled correspondence. It is a mechanism for a generic drug manufacturer or related industry to request information on a specific element of generic drug development and for the agency's direct, brief, and timely response. As it relates to response time, goal dates are outlined in the GDUFA commitment letter, which is generally 60 days, but could be up to 120 days for certain controls. A controlled correspondence is suitable for products where Q1, Q2 sameness is required by the regulations. Also, it is for products where Q1, Q2 sameness is not required by the regulation, but recommended in a guidance. For example, a product-specific guidance may recommend a specific bioequivalence approach. Please note that controlled correspondences will generally be routed to the Division of Filing Review, and in some cases, regarding complex drug products, it may be routed to the Office of Research Standards. FDA recommends that when submitting a controlled correspondence, it includes information about the RLD, such as the application holder, the application number, proprietary name, active ingredient, strength, and to specify the fill volume of parenteral drug products as it relates to strength, the dosage form, route of administration, approval date, and marketing status on whether the product is a prescription, over the counter, or in the discontinued section of the Orange Book. All these items can be found in the Orange Book. As a reminder, if a requester is seeking formulation assessment for multiple drug products, FDA recommends that each drug product request be submitted in a separate controlled correspondence. For instance, requests for Q1, Q2 formulation assessment for drug products with different RLDs, or requests for Q1, Q2 formulation assessment for drug products with multiple strengths, because each strength is a separate drug product. FDA recommends that no more than 3 proposed Q1, Q2 formulations of a single product be submitted in one controlled correspondence. Limiting a single control correspondence to no more than 3 formulation requests allows for FDA's targeted and timely review of such requests. In addition, the agency recommends against submitting a request for a Q1, Q2 formulation assessment and a separate request for evaluation of a proposed inactive ingredient amount or concentration at the same time. FDA does not intend to review proposed formulations for which Q1, Q2 sameness is not required by the regulation, nor is it recommended as part of a bioequivalence approach and a guidance. Formulations that are not Q1, Q2 the same as the RLD are permissible for certain products that are not parenteral, ophthalmic, or otic product, provided that the differences are identified and characterized with information demonstrating that the differences do not affect the safety or efficacy of the drug product. The acceptability of such differences would be considered in the context of an ANDA review. Please note, the agency is limited in the amount or type of information that FDA may disclose in response to a request for Q1, Q2 sameness assessment because formulations generally are trade secret information, pursuant to 21 CFR Section 20.61a. FDA does not intend to provide clarification on why a formulation is not Q1, Q2 the same as the RLD. In certain circumstances, a controlled correspondence may not be the optimal mechanism to gain FDA's feedback. The pre-ANDA program is intended to clarify regulatory expectations for prospective ANDA applicants early in product development, assist applicants in developing more complete submissions, promote a more efficient and effective ANDA assessment process, and reduce the number of assessment cycles required to obtain ANDA approval. For example, a product development meeting for complex products may provide a better forum for questions related to an ANDA development program and to seek dialogue with the agency on a particular matter. The material submitted in support of a meeting request should help the agency determine whether a meeting is appropriate, and the prospective ANDA applicant should have enough knowledge of the complex drug product to allow FDA to provide appropriate feedback that will advance product development early in the process. For instance, the prospective ANDA applicant has generated its own data to be discussed. And such information would allow for a forum for scientific exchange on specific issues, such as a proposed study design, an alternative approach, additional study expectations, or questions in which FDA agreed to provide targeted advice regarding an ongoing ANDA development program. It should be noted that complex products are outlined in the Guidance for Industry on formal meetings between FDA and ANDA applicants of complex products under GDUFA, and generally include products with complex active ingredients, formulations, routes of delivery, dosage forms, drug-device combination products, and other products where complexity or uncertainty concerning the approval pathway or possible alternative approach would benefit from early scientific engagement. For further information, please refer to the Guidance for Industry, the GDUFA 3 Commitment Letter, or the Manual of Policies and Procedures 5220.8 on evaluating requests for and conducting product development and pre-submission pre-ANDA meetings. A product development meeting would be suitable for questions regarding 1, development of a complex generic drug product for which FDA has not issued a product-specific guidance, or 2, an alternative equivalence approach for a complex product for which FDA has issued a product-specific guidance. Where a controlled correspondence response would not adequately address the prospective ANDA applicant's questions, and that a product development meeting would significantly improve ANDA assessment efficiency, such as ultimately decreasing the number of review cycles for the application. Furthermore, the prospective ANDA applicant's meeting package should include any data generated and specific proposals for product development. For instance, details regarding the proposed product development plan, such as an alternative study design and sufficient justification to support the proposal, should be provided. FDA can meet the product development meeting goal by either conducting a meeting or providing a meaningful written response that will inform drug development and/or regulatory decision-making to the prospective ANDA applicant within the applicable goal date. FDA recommends that the prospective ANDA applicant submit no more than one request for a product development meeting for the specific complex product per year, but FDA anticipates that some prospective ANDA applicants of complex products may request more than one product development meeting. After product development meetings, if a prospective ANDA applicant is seeking further clarification or has new questions related to what was discussed at the meeting, FDA recommends that the applicant submit such a request with any new information or data in a controlled correspondence. However, if the new information, data, or questions for agency input will not be adequately addressed in a controlled correspondence, the prospective ANDA applicant can request an additional product development meeting. FDA will determine whether to grant this subsequent product development meeting based on the content of the meeting request and the meeting package and available resources. For further information, I recommend that you read the Guidance for Industry on Controlled Correspondence related to generic drug development, the Guidance for Industry on Formal Meetings between FDA and ANDA applicants of complex products under GDUFA, and/or the Manual of Policies and Procedures 5220.8 on evaluating requests for and conducting product development and pre-submission pre-ANDA meetings. Thank you for your participation. We will now turn it over to the Q&A session with panelists.
Okay, good afternoon, everyone. We're in the home stretch in the last session. I know it's late for some of our participants. So here is when I invite our panelists to join us on video so that we can have a Q&A session. I guess we'll dive right in. I think one of the first questions that we have will be directed to Elizabeth. For control correspondences, for Q1, Q2 formulation assessments, does the agency evaluate the active ingredient along with the inactive ingredient?
Hello. Could you repeat the question?
Oh, yes, Elizabeth. For control correspondence Q1, Q2 formulation assessments, does the agency evaluate the active ingredient along with the inactive ingredient?
Okay, thank you for the question. The agency does not assess the active ingredient of the drug product during the controlled correspondence review of a Q1, Q2 formulation assessment. Q1, Q2 evaluations of proposed formulation include only the inactive ingredients. Thank you.
All right, thanks. So the next question is for Julia. One of the questions is, to the extent a pH adjuster is used to convert the active pharmaceutical ingredient to form the API salt, the pH adjuster is not included in the Q1, Q2 assessment because it becomes part of the active ingredient. And if excess pH adjuster is used, that excess amount is generally considered an inactive ingredient subject to the Q1, Q2 assessment. How should an applicant list the use of pH adjuster in such circumstances?
Hi, Vin. Thanks for that question. So we recommend that an applicant clearly lists the amount of the pH adjuster separately based on the different functions intended in the drug product. For example, if the pH adjuster is going to be used to convert the API, then place that as one line item identifying the specific amount, and then separately list the pH adjuster amount used to adjust the pH.
Great, thanks for that, Julia. So moving on, I'm going to try my best to group these questions together, but I might be jumping a little bit over the place regarding the different topics here. So bear with me. So the next question is for Melissa. Would differences in the exception excipient, such as preservative, buffer, tonicity agent, or thickening agent in ophthalmic drug product be acceptable?
Hi, thanks for the question. Yes, so differences in an exception excipient is acceptable for purposes of meeting the inactive ingredient requirements at 31494 A9. However, if, for example, an ANDA is concerning or involving seeking approval for an ophthalmic drug product where, it has to be Q1, Q2 the same to the RLD, and generally, as a scientific matter, the FDA expects that it's QQ same with respect to all of the inactive ingredients, including the exception excipients. If there's a difference in one of those exception excipients, while that's permissible in terms of the inactive ingredient requirement, where those differences exist, the agency would expect additional submission of appropriate BE studies to support that difference. And so, generally in our PSGs, I believe that we've been recommending in vivo, submission of in vivo BE study to support the difference in exception excipient for ophthalmic drug products where it's noted. But keep in mind that PSGs are recommendations, they're not requirements. And so to the extent that you may be considering an alternative approach that's not an in vivo, and it doesn't preclude you from using that approach, but we would encourage you to reach out to the agency to have that discussion before you start your study to just ensure that, what you're planning to submit would, the agency would consider, acceptable and would meet the statutory and regulatory requirements. Thanks.
Thanks for that, Melissa. Okay, so the next question I have is for Elizabeth. Could you summarize what information is recommended to be included in an information table for Q1, Q2 assessment inquiry?
Thank you for that question, Vin. Please include function, hydration state, grade, purity, viscosity, all units of measurement as applicable. The description of each excipient can be included within the ingredient column of the formulation table, or you could utilize a footnote to describe an excipient. For a parenteral drug product, please include the fill volume of the drug product. And also please note for co-packaged drug products, it is important to include a drug composition for each packaged component. Thank you.
Thank you. So that brings me to my next question for you, Melissa. One of the questions is it possible to view an example of a control correspondence, as in what will represent a high quality control correspondence?
Thanks for the question. So control correspondence is considered confidential correspondence between an applicant and FDA on that applicant's specific generic drug development. And so since it's confidential information, we're not able to share an example of what would be considered a high quality control. But I would encourage you to just, take into consideration the recommendations we're making for you here today in this session to help you ensure that that your control would be of high quality. So for example, like including a clear description of the RLD product that you plan to use as the basis for your submission, to make that QQ assessment for your proposed drug formulation. And to also include clear, detailed information about your proposed formulation with an accurate description, units of measurement, and any appropriate footnotes as needed, to help ensure that the agency has all of the information that we would need to be able to make a QQ assessment against the RLD. Thanks.
All right, thanks for that, Melissa. The next question we have is for Julia. Could we include separate elements of a generic drug development question, no more than 3 questions for a single drug product in a single control correspondence?
I need to unmute myself. Thanks for the question, Vin. So if a requester seeks information related to separate elements of a drug, generic drug development, the agency recommends that the requester submit separate control correspondences for separate elements. No more than 3 proposed formulations of a single drug product should be submitted in one control correspondence, and limiting a single control correspondence to those assessment requests, it allows for the agency to respond to the control correspondences in a targeted and timely manner. Thanks for the question.
Awesome, thanks for that. Okay, so we'll pivot a little bit. The next question is for Bryan. How many formulations can I submit in a product development meeting?
Yeah, thanks for that question. So just as Julie was mentioning, for controlled correspondences, generally the maximum that's requested to be submitted is around 3. So in terms of the product development meeting, and generally we recommend that the number of formulations be comparable to that. So 3 would be a good submission number.
Thanks, Bryan. I think another question related to product development meetings is, how would I get feedback from the agency if there's no product specific guidance? And what if the drug product is not complex?
Yeah, thanks for that. I can take this one. So as we mentioned earlier, the FDA can provide feedback on the proposed bioequivalence approaches through the product development meeting process. And this can include the evaluation of test formulations if the firm is actually proposing formulation sameness as part of the bioequivalence approach. So the end of meeting request process provides a way of evaluating or receiving feedback from the FDA for a bioequivalence approach when there's no product specific guidance available. Now, in the terms of a non-complex product, generally the requirements for Q1, Q2 sameness are dependent on the product itself. If it's in an injectable or ophthalmic or otic solution, for instance, then these products are generally required to be Q and Q2 the same. So then a control correspondence could be used in place of submitting any type of meeting request. Now, if it's another non-complex product that doesn't have this type of requirement, then in general, the product development meeting request isn't going to be applicable in this case, but we could grant this, of course, is at the discretion of the FDA if we were to feel like this meeting would be beneficial to the efficiency for the review for this product.
Great, thanks for that, Bryan. Okay, we're gonna kind of flip back to control correspondences because I think most of those questions are related to controls. So the next one is for you, Melissa. When submitting a Q1, Q2 assessment control inquiry, do we need to list functions for all ingredients listed in the composition table?
Thanks for the question. So the short answer is yes. The agency wouldn't be able to give you a full assessment of the, in terms of, whether your product is QQ same to an RLD, per the requirement under 31494A9. When there is an omission of the function of the ingredient in the formulation during, review in the control correspondence pathway. So hence, especially because we're not receiving, all of the other additional information that is normally submitted as part of a full application package. And so the function becomes a very important part of the generic drug product formulation information that the agency would need to be able to give you a QQ assessment of your formulation against the RLD. And that should be listed for all active and inactive ingredients. Let's see, and with respect to parental drug products and ophthalmics and audit drug products, right, there's a limited enumerated list of excipient types that are considered exception excipients. And so for the purposes of determining whether the inactive ingredient requirement at 314.94A9 applies or not, right, we need to be able to assess whether you're identifying an ingredient that would qualify as one of these noted exception excipients. Thanks.
Great, thanks for that, Melissa. So the next question actually relates to function as well. So when a pH adjuster has dual functions, such as pH adjuster and a buffer, and whether we treat this inactive ingredient as a buffer, which is an exception excipient, or a pH adjuster non-exception, is dependent on its main function. Is this understanding of the guidance correct?
The short answer to that is no. So when a pH adjuster functions as both a pH adjuster and as part of a buffer system, we will generally apply the function of the inactive ingredient as identified and demonstrated by the applicant or the application holder. If you as the applicant identify the inactive ingredient as a pH adjuster, then FDA will consider that ingredient to be a non-exception excipient subject to Q1, Q2 sameness. On the other hand, right, if you identify that ingredient is functioning as a buffer, right, then it is considered an exception excipient. Now, I think it's important to note, though, however, that like where an applicant, like let's say the RLD notes that the ingredient is a pH adjuster and the test applicant wants to note that it's a buffer. And there's that difference in how that function is applied to that same ingredient as listed in each respective formulation. The agency will, in a way, like default to the non-exception excipient and treat that ingredient as a non-exception excipient because one of you has listed it as a pH adjuster rather than a buffer to make it a non-exception excipient. Now, with that said, 314.99B waiver does allow for flexibility in that regulatory framework, as we've discussed today, especially with respect to the pH adjuster difference, where there is a buffer also listed, as another inactive ingredient in your formulation. And so I'd refer you back to I believe it was the previous session or 2 sessions ago, where they discussed those scientific principles, to help support coming up with the scientific rationale to include as supportive justification for such a waiver request. Thanks.
Great, thank you for that, Melissa. So the next question is for Elizabeth. And so if the AND is a lyophilized product and use some solvent or some solvents, should we include in the QQ composition table?
Thanks for the question. Yes, we recommend to include the solvent in the proposed formulation table. However, any solvent included in the proposed formulation table that is not in the final drug product, it should be noted as such. For example, if a lyophilized product formulation table included water, and water is not in the final drug product, it is important to indicate that water is removed during lyophilization of the drug product. Thank you.
Okay, thank you for that, Elizabeth. So the next one, speaking of whether to include a particular inactive ingredient or not, what about, we have a question here that kind of lines up with that, is for for Q1, Q2 control correspondence inquiry, do we need to submit a composition for each of the package components?
Okay, let me mute. Okay. Okay, yes. Yes, it is important to include drug composition for each packaged component for co-packaged drug products. In other words, if a drug product comes in a dose pack, such as a vial containing lyophilized powder and diluent, please include formulation for both the lyophilized powder and diluent. It is important to provide compositional breakdown, both qualitative and quantitative, for all components of a drug product.
Okay, thank you for that, Elizabeth. So the next question is for Melissa. Is submission of a product development report part of a control, should be submitted as part of a control in case when seeking a formulation assessment?
Thanks for that question. As an applicant, you are welcome to include reports from a product development meeting that you've had with the agency if you think that that information would be supportive of the question that you're asking in your control. But submission of that information from the product development meeting isn't necessary to obtain a formulation assessment for QQ sameness to the RLD. So for example, like if you have like a complex drug product where the excipients that are included in that formulation are complex in and of themselves, and you think that the information that was discussed in your pre-development meeting would support, a finding, for example, of like a Q1 sameness, and to the extent that you think that it does support that, and you want to include it, you're more than welcome to, and we would take that under consideration in our in evaluating your formulation and formulating our response back to you. Thanks.
Okay, thanks for that, Melissa. So the next question is for Julia. Is the filling volume required in a QQ formulation when submitting a control correspondence for a rectal suspension? Should the filling volume be the same as the RLD?
Thanks, Finn, for that question. So since rectal products aren't required to be QQ same to the reference listed drug, this would be a control correspondence that wouldn't be accepted by the agency as a control correspondence in the QQ space.
Great. Thanks for that, Julia. So since we're talking about fill volumes, the next question is for Elizabeth. For parenteral drug products, can a Q1, Q2 formulation assessment inquiry is submitted as a control contain multiple fill volumes if the formulation is the same for all fill volumes?
Thank you for your question. The agency recommends that formulation assessment for multiple drug products be submitted in separate control correspondences. This includes separate formulation assessment requests for drug products with multiple strengths, such as parenteral drug products with different fill sizes, as each fill size or strength is a separate drug product. And this information is found in Guidance for Industry Control Correspondence Related Generic Drug Development, published December 2020. Thank you.
Great, thanks for that, Elizabeth. So the next question is for Melissa. So when submitting a control correspondence, does the applicant need to submit certain characterization data to support the proposed excipient concentration with the RLD data in the control correspondence?
Thanks for the question. So an applicant may include characterization data to the extent you think that it would support that the ingredient is Q2 same to the RLD. If you want to include that in a control, that's fine, and you're more than welcome to. However, please note that with respect to the agency's assessment of a formulation that's submitted as a control, the response that you'll be receiving back from the agency is going to be limited to indicating whether your formulation is actually QQ same to the RLD or not, right? Now, on the other hand, if you are planning to use this characterization data to support and justify the concentration that you're using in your proposed formulation that may be different from the concentration used in the RLD, that information would be assessed during the scientific review of your ANDA, and wouldn't be assessed during a control. Thanks.
Great, thanks for that, Melissa. So the next question is for Elizabeth. So the question is, when evaluating Q1, Q2 status for a parenteral drug product, if the parenteral requires sparging with nitrogen or nitrogen headspace, how is the nitrogen evaluated as the quantities can hardly be known?
Nitrogen used as a processing aid or a component used to fill headspace is not evaluated as a part of Q1, Q2 assessment. Thank you.
Awesome, thanks, Elizabeth. So the next question is for Julia. What is the importance of including the hydration state of an ingredient, when submitting formulations for FDA's feedback?
Thanks, Vin, for the question. So including the hydration state is very important because, especially in the situations where there are certain excipients that have different hydration states, this can impact the determining whether or not the proposed product is quantitatively the same as the reference listed drug. And so, and since the regulations require parenterals, ophthalmics, and otics to be both qualitatively and quantitatively the same, it is necessary for the control correspondence to include the hydration state in order for the agency to conduct a complete review of the control correspondence and provide adequate information to the applicant.
Awesome, thanks for that, Julia. Excuse me. So the next question is for Elizabeth. What types of issues prompt the agency to send an information request for control correspondence? I think you're on mute there, Elizabeth.
Sorry about that. An information request is sent by the agency in the course of reviewing control correspondence when more information is needed to conduct assessment. When reviewing a Q1, Q2 formulation assessment inquiry, omission, discrepancy, or ambiguity in the formulation table can trigger an information request. The agency may send information requests for omission of function, unclear units of measurement, missing hydration state, missing total fill volume of a parenteral drug product, and also for missing diluent. Thank you.
Great, thanks for that, Elizabeth. So the next question is for Julia. If we received a control correspondence Q1, Q2 formulation assessment response from the agency that the proposed formulation is acceptable, could we assume that each inactive ingredient is also reviewed for maximum quantity allowance?
That's a great question, Ben. So when an applicant submits a control correspondence for a Q1, Q2 formulation assessment, we at the agency do not consider the inactive ingredients allowance evaluation. So if an applicant submits a control correspondence for QQ evaluation, the determination of the inactive ingredient maximum quantity allowance is not assessed. Therefore, if an applicant does want information from the agency about the maximum quantity, then that should be submitted as a separate controlled correspondence. Thanks, Finn.
Awesome. Thank you for that. The next question we have is for Elizabeth. So could we submit a clarifying question in response to a Q1, Q2 formulation, in response to a Q1, Q2 formulation control correspondence response?
Yes, FDA will respond to requests to clarify ambiguities in the agency's control correspondence response. The request for clarification should be submitted within 7 calendar days of issuance of FDA's control correspondence response. The scope of the clarifying questions should be limited to the content in the response. But please note, FDA does not intend to provide clarification on why a formulation is not Q1, Q2. Any requests to review follow-up questions or new or additional information will be considered a new control correspondence.
Great, thank you for that, Elizabeth. So the next question we have is for Melissa. Given the long development timelines for complex injectables, how can we request the agency to reconfirm accuracy of the composition in the RLD package insert / label?
Thanks for the question. So based on the presentations today, we're aware that there have been instances where, the information wasn't, and, it turned out the information wasn't accurate in the RLD labeling and was later corrected. However, right, for you as an applicant, we would recommend that you begin with generally a presumption that the information contained in the RLD labeling is accurate to the RLD composition of the drug product, to the extent that the RLD labeling is required to disclose that information per the labeling requirements at 201-100B5. Right, and just keep in mind that there are certain pieces of information related to a drug's formulation that's not required to be disclosed in RLD labeling. And as such, that information on RLD labeling alone may not be sufficient to achieve a QQ match to the RLD. Unfortunately, FDA is not able to comment on requests from ANDA applicants to confirm or reconfirm whether the information in RLD labeling is accurate. However, if you have verified your reverse engineering methods and repeatedly are obtaining results that appear to conflict with the information that is listed in the RLD's labeling, we'd encourage you to share that with the agency as part of a control, asking for a QQ assessment with consideration given to the potentially conflicting results that you're obtaining in your own reverse engineering efforts, for the agency's consideration. The agency is continually working to improve our review processes and monitoring of our FDA-approved products. And where there is a discrepancy, where we do find a discrepancy, we will take measures to make sure that those issues are addressed with respect to the RLD's labeling. So yeah, thank you for the question.
Thanks, Melissa, for the elaborate response. The next question is more procedural. So Julia, which office or division reviews the Q1, Q2 formulation assessment request for control correspondences?
Thanks, Vin, for that question. So the majority of control correspondences are responded to by the Division of Filing Review within the Office of Regulatory Operations. They look at the inquiries that have to do with non-complex drug products. However, if the agency has determined that the drug product is complex, then the Office of Research and Standards will review the control correspondence inquiry. Thanks, Vin.
Great, and it looks like we have time for one more question. Melissa, and this one's for Melissa. Is there a specific guidance for hybrid generics? In other words, where the route of administration is significantly different, and so sameness with regard to formulation and efficacy will be different?
Hi, thanks for the question. So when we're talking about whether it's permissible to submit that type of drug product as an ANDA, a 505(b)(j) application, remember that any change in route of administration, dosage form, or strength, or for combination drug products where one of the active ingredients is different than the referenced product, any of those types of changes would actually require you to submit a petition for making that change from the reference listed drug before, and having that petition approved before you would be eligible to submit such an application as an ANDA to FDA for approval. Your petition for such a change should include justification And for example, for where a petition involves a change in route of administration, should include supportive information for why that difference from the RLD in route of administration should be considered permissible to be reviewed and approved as an ANDA. And I would refer you to 31493, 21 CFR 31493 for additional information on how to make such a petition. And we'll also note that if there is another NDA product that is already approved that covers the change that you are seeking to petition for, FDA will deny a request to petition for such a change as an ANDA. The requirement there would then be for you to submit an ANDA citing that NDA as the basis of submission, the NDA that contains the change you're trying to petition for as the basis of submission for your drug product. Thank you.
Great, thanks for that. That was very informative. So I want to take out a moment to appreciate everyone's time and effort in putting this webinar together and for your participation today. So with that, I hope that you find it informative. And so with that, I turn it over to Dr. Bryan Newman for closing remarks.
Yeah, so yeah, thanks. I have the privilege of closing out today's webinar. So since we've reached the end, I want to first thank the CRCG and our FDA colleagues, particularly the webinar planning committee, including Dr. Anubhav Kavaratna, for their focus and dedication for coordinating and organizing this webinar. I want to thank all of our speakers from both the FDA and industry for providing very informative and detailed presentations on the many complex considerations related to formulation assessments. I also want to thank our panel members for sharing their time and expertise to address the audience's questions and provide interesting discussions on these topics today. And lastly, I want to thank all of you for the active participation during our several question and answer sessions. So throughout this webinar, we've received many questions here from the audience through our chat box, and we've made our best efforts to try to address as many as we could today in the webinar. So if you feel that if we were not able to address your particular question, or you have a discussion topic that wasn't touched upon during this webinar, I want to please mention to take the opportunity to provide your thoughts in our post-webinar survey that will be available in the coming days. My hope is that this webinar provided an opportunity for you all to learn about the regulatory framework that supports FDA's approaches for evaluating formulation sameness and all the considerations that go into this assessment across the different dosage forms and the routes of administration. So looking back over what was covered today in session 1, we heard 3 presentations that covered the regulatory framework related to inactive ingredients and formulation sameness for the different dosage forms and routes of administration. Formulation sameness often plays an important role in establishing bioequivalence for different drug products, where some require formulation sameness based on the regulations, as we heard, and while others, this is part of their bioequivalence recommendations that are provided in FDA's product-specific guidances. So our speakers also discussed the reasoning for how FDA responds to formulation assessment requests, as well as several scenarios that generic applicants can encounter when you're selecting the right RLD or reference standard for generic development, and ways to get around these challenges. In session 2, our speakers provided 2 presentations that discussed the considerations involved when FDA performs its assessment of qualitative and quantitative sameness for formulations and active ingredients, as well as a presentation on the current thinking involved when making a no significant difference assessment with topical formulations. While the Q1, Q2 sameness assessment can sometimes appear to be a rather straightforward determination, often this assessment poses more of a challenge because these formulations, particularly for more complex dosage forms, contain inactive ingredients where the sameness determination presents a scientific or analytical challenge. So our speakers provided insights into FDA's thinking for how these challenges can be addressed and how generic applicants can include the necessary information for FDA to assess sameness without having FDA needing to submit an information request. Now, session 3, we heard 2 presentations from FDA on the considerations surrounding safety of an inactive ingredient in a generic formulation, as well as a presentation on the inactive ingredient database, which can inform generic applicants on the acceptable exposure levels for inactive ingredients in an FDA-approved drug product. As these speakers illustrated in their presentation, the development of a generic formulation involves not just the consideration of the inactive ingredients' impact on product performance, but also the consideration for how the levels of inactive ingredients can influence the safety profile related to the RLD, particularly between adult and pediatric populations. Now, in session 4, this provided us an opportunity to hear from our industry representatives on some of the challenges faced when developing a generic formulation, both from the scientific as well as the regulatory side. This was followed by an FDA presentation that provided insight into the agency's current thinking on pH adjusters, including a walkthrough of several examples of how a generic applicant may work to support a potential Q1 and Q2 difference with the pH adjuster in a test formulation as compared to its RLD. The industry feedback on their challenges with formulation development and the formulation assessment process is valuable for the agency since it helps to highlight specific scientific challenges where more research may be needed, as well as areas with the assessment process that could benefit from greater efficiency and guidance. As illustrated by the pH adjuster guidance, FDA is continuing its efforts to provide more streamlined approaches for generic development. And finally, we've heard from 2 FDA speakers on the best practices when submitting a formulation assessment request in a controlled correspondence or a meeting request, as well as some of the common reasons information requests are sent to applicants and ways that to avoid these. A key take-home message from these presentations was that submitted formulation tables need to be sufficiently detailed and without ambiguity for the agency to evaluate these in an efficient manner. So when submitting a formulation table for assessment, applicants should carefully consider the appropriate information that should be included for each inactive ingredient. Otherwise, the agency's response can be delayed if one or more information request is needed before comparison between the proposed formulation and the RLD can be completed. So overall, I believe the presentations throughout this webinar have done an excellent job of demonstrating the wide range of scientific and regulatory considerations surrounding a formulation's inactive ingredients and the challenges involved with showing that a given test formulation meets FDA standards for sameness related to the inactive ingredients. Again, I encourage the audience to provide your input on what you learned from today's webinar and what you think should be included in future workshops for inactive ingredients and formulation assessments in the post-webinar survey that will be available in the coming days. And with that, I thank you all for attending today's webinar and I hope you have a good rest of your day.